Hello dear students, this is Dr. Adwet and welcome to lecture number 14 of this chapter called as Cell, the Unit of Life. So this is the last lecture about the last part of this chapter called as Cell, the Unit of Life. Before we start with today's session, there are a few things that I would like to discuss with you. Number one, if you enjoy today's session and if you understand everything that we have discussed in today's lecture, please do not forget to hit the like button given below. It lets me know that you have understood everything and that you have enjoyed today's session. At the same time, if you would like to see more such content regarding standard 11th and 12th NCERT biology, do not forget to subscribe to our channel, which is Dr. Adwet's NEET Biology. Also, while you're down there, please hit the bell icon, which will inform you immediately about all my future uploads. Also, if you have any questions or any doubts regarding anything related to biology, please leave a comment below in the comment box and I will try and reply to you as soon as possible. Also, do not forget that each and every lecture has its own assignment. You will find the description for this assignment in the description box given below. Please find the link, download the assignment and solve it immediately after seeing today's video. It will help you to consolidate your knowledge about today's session. And lastly, if this video has been useful to you, if it was valuable to you, please share it with all your friends whom you believe will benefit from this lecture series. So with all of this out of the way, let us see what we have done in the chapter of cell in the previous 13 lectures. So let's have a quick recap. So in lecture number 1, we did introduction to the cell. In lecture number 2, we saw history of microscopy and the cell theory. In lecture number 3, we spoke about parts of cell, basic genetics and characteristics of a cell. In lecture number 4, we spoke about the nucleus. In lecture number 5, we spoke about plastids. In lecture number 6, we spoke about mitochondria. In lecture number 7, we spoke about ribosomes. In lecture number 8, we spoke about the Golgi bodies, lysosomes and vacuoles. In lecture number 9, we spoke about cytoskeleton. In the 10th lecture, we spoke about the plasma membrane, and in lecture number 11, we spoke about the cell wall. So for the first 11 lectures, we discussed the eukaryotic cell in detail. Those are the first 11 lectures. Now, in lecture number 12, we spoke about prokaryotic cell and we finished off the prokaryotic cell and differences between the eukaryotic cell and the prokaryotic cell in lecture number 13. So lecture number 12 and 13 were all about the prokaryotic cell. So this is what we have done, dear students, from lecture number 1 to lecture number 13. Which brings us to today's lecture. So if we finished everything about the cell, what is the bit that is remaining? So if we consider a eukaryotic cell, as far as its nucleus is concerned, it shows the presence of something called as chromosomes. And the last part of this chapter in the NCERT discusses those chromosomes and their types. Now, I did not teach this to you when we were doing the chapter of cell because you had no idea about chromosomes, packaging of chromosomes, all of which I have explained to you when we did the chapter of cell division. So now that we have finished the chapter of cell division, we can come back to this chapter of cell and finish off details regarding the chromosomes. So before you see today's session, it is extremely important that you revise or go through the lectures which are now going to show up on your screen. So you will have to see Cell, the Unit of Life, lecture number 3 as well as 4. In fact, what is more important is going to be lecture number 4 of the chapter called as Cell. And please have a look at lecture number 1 to 4 from cell division. So this is what I would like you to do before you see this lecture. So please go through lecture number 3 and 4 of the chapter called as cell and lecture number 1 to 4 of the chapters called as cell division. Only when you are thorough with that, or you are sure that you remember everything that we have discussed in those lectures, should you embark upon today's lecture. Or else pause the video right now or close this video, go back to those lectures, and I will leave a link for all those lectures in the description box below. So if you want to have a quick revision, you can just go to the links in the description and see those videos before you come back to this video and see it from this point on. So I'm assuming that now these lectures are clear with you, and if that is the case, we can start discussing chromosomes. So what exactly are chromosomes? This is something that you have done in lecture number 4 of cell, and along with that we have also seen it in the lectures number 1 to 4 of cell division. So you know that chromosomes come from the Greek word chroma, which means color, and soma, which means body. So colored bodies which are seen in the nucleus during cell division are called chromosomes. So chromosomes are filamentous bodies present in the nucleus. They are visible only during cell division after various staining procedures. Another point to remember here is they are visible only during cell division after staining procedures. You cannot see chromosomes by directly placing a cell in the nucleus. You will have to use staining methods. Okay. Chromosomes have the ability of self-replication, meaning that they can make copies of themselves, and they play an important role in heredity, variation, and evolution of species. Chromosomes which are morphologically and genetically identical are called homologous chromosomes, and you know about this in great detail because we've done all of this in cell division. So homologous chromosomes are those which are going to be the same in their size, in their structure, in their centromere position, and in their gene locus. They will have similar genes, but the alleles may or may not be the same. So all of this already discussed. Now, what about the size of chromosomes? The size of chromosomes varies from species to species. If we speak about human beings in general, the metaphase chromosome. So here is why it is important to know about cell division, because we are using the word metaphase. So you know that the M phase in cell division comprises of prophase, metaphase, anaphase, and telophase. Prophase is when the chromosomes actually start to appear within the nucleus. Metaphase is when they have maximum condensation. Anaphase is when they split into the daughter. The sister chromatids are going to split, and then you are going to have telophase when the chromosomes with the single chromatids are going to go towards the opposite poles, and then are going to again form the chromatin network. So each metaphase chromosome varies from 0.1 to 33 micrometers in length, and it can be anywhere from 0.2 to 2 microns in thickness depending on the species. Okay, what about the shape of the chromosomes? So the shape varies according to the stage of cell division, and by the time we end today's session, this point will become clear to you that the shape varies according to the stages of cell division. In interphase, the chromosomes are in the form of chromatin network, as I told you, and they show maximum condensation in the metaphase. So prophase is when they start to appear in the nucleus. In the metaphase, they are going to be at maximum condensation, and they will appear the most clear during metaphase along the equatorial plate. In anaphase, they will split, so you will now have chromosomes with single chromatids instead of chromosomes with sister chromatids. And then of course in the telophase, when they are at the opposite poles, they will then again form the chromatin network. So they will decondense and form the chromatin network. So in interphase, chromosomes are in the form of chromatin network, while in metaphase they show maximum condensation and appear short and thick. All of these things mostly you are aware of when we have done cell division. Now comes the important point for which I am taking this special lecture. What about the structure of the chromosome? So a few things you already know, and there are a few things which will be new to you. Let's see. So what you see here is something that you have seen multiple times during the chapter of cell division. This is a metaphasic chromosome, and it is showing the two sister chromatids which are joined together at the centromere. These are things which you already know. So a metaphasic chromosome has two identical halves which are called as sister chromatids, and each chromatid in turn is made up of subchromatids called chromonemata. So if you see the diagram, you will notice that this here is a chromatid. This is a single chromatid. Now this single chromatid is going to have two parts. There is this part here, there is this part here, there is this part here, and there is one more part here. So these parts are the subchromatids, which are called as the chromonemata or the chromonema. Singular is chromonema and plural is chromonemata. So each chromatid in turn is made up of subchromatids called as chromonemata. Singular will be chromonema. This is something which you have not learnt before. So there you go, chromonema. The chromatids lie side by side and are held together at one point called as centromere, which you already know. And on either side of this centromere are disc-shaped structures called kinetochores. So you know that here is the centromere in the diagram. I hope you can see that, the black part, that is the centromere. And next to the centromere, what you see in orange, are the kinetochores. So from the chapter of cell division, you know that during cell division, the spindle fibers are attached to the kinetochores. The spindle fibers are attached to the kinetochores. The centromere is also called as the primary constriction. So the basic idea is that if you were to see a chromosome, you will notice that the area where the centromere is present, there is a slight constriction. एक खांचे जैसा है. So that area where you have constriction, here the main one will be the centromere. So the centromere can also be called as the primary constriction. Now this is very clear and apparent when you speak about a telophasic chromosome. So children, pay attention now. This, what you see on your screen right now, this single diagram that you see is the metaphasic chromosome. And what you see now is going to be the telophasic chromosome. So here you have the metaphasic chromosome and here you have the telophasic chromosome. What is the difference between the metaphasic and the telophasic chromosome? During metaphase, the sister chromatids are attached to each other with the centromere. But in anaphase, the sister chromatids will separate, and in telophase, this chromosome that has now appeared on the screen, this chromosome, this will only comprise of a single chromatid. It will comprise only of a single chromatid. So now if you see this telophasic chromosome, it will be very clear to you that the area where the centromere is constricted, and hence that area where you see the constriction is called as the centromere or the primary constriction, the main constriction. So the centromere is also called as the primary constriction. Besides the primary constriction, additional narrow non-staining, so they appear to be transparent or clear without any color in the compound microscope. So besides the primary constriction, additional narrow non-staining areas are present, which are called secondary constrictions. So if you see this diagram, these pictures which you see are going to be called as the secondary constrictions. So primary constriction is going to be the centromere, whereas these areas are going to be the secondary constrictions. So if you see the telophasic chromosome, if you see the diagram on your screen right now, the telophasic chromosome, this what you see here is the main constriction. You can see that it's the biggest constriction on this chromosome. So this is called as the primary constriction or the centromere, whereas this one here, and to a certain extent this one here are called as secondary constrictions. So primary and secondary constrictions. Now the secondary constrictions are sometimes also called as the nucleolar organizers. Not nuclear, nucleolar. Now they are called nucleolar organizers because they help or are necessary for the formation of nucleolus. So we have done that before when we were talking about the nucleus in lecture number 4 of cell. So as far as the nucleolus is concerned, its formation requires help from the secondary constrictions. So they are also called as nucleolar organizers. So secondary constrictions can also be called as nucleolar organizers. So some of the secondary constrictions are called nucleolar organizers because they are necessary for the formation of the nucleolus. the part or the fragment of the chromosome beyond the secondary construction. So if you were to see the diagram here, and if you were to see this as the secondary construction, this is the fragment, especially this part which they have labeled here. This is the fragment which is beyond the secondary construction. Now, sometimes those small fragments which are beyond the secondary construction, they are also called as satellites. So the part or fragment of the chromosome beyond the secondary construction is short, spherical, and is called as the satellite. Lastly, that brings us to the very edge or tip of the chromosome. The very tip of the chromosome, which is called telomere. The tip of the chromosome is called as the telomere. And the unique property of telomere, so in this case, this is the tip which will be called as telomere. So if you see the metaphasic chromosome, the one in blue, here you have the telomere. This can be called as the telomere. So the unique property of these telomeres is that they are the tips of the chromosomes and they do not allow the sister chromatids to stick to each other. So if this is the chromosome, it will not allow this to happen. It will not allow the tips of these sister chromatids to attach to each other. However, the other unique property is that they will help the tips to attach to the nuclear membrane. So telomeres help an attachment of the chromosome to the nuclear membranes or the nuclear envelope, and they do not allow the tips of the sister chromatids to attach to each other. So remember this unique property about the telomere. Telomeres have a unique property in that it prevents the ends of the chromosomes from sticking together. So the sister chromatids along with the neighboring chromosomes, they will not be able to attach to each other or stick to each other. But it helps to attach to the nuclear envelope. And lastly, the surface of the chromosome has small swellings which are called chromomeres. So if you will see this part of the chromosome here, please bring your attention to this part of the chromosome. You will notice that there are these small elevations that you may see on a chromosome. Those little foldings or elevations that you can see are going to be called as the chromomeres. So to scientists who study chromosomes, these chromomeres are important because these chromomeres or little elevations help them to position where genes are located. So they help them to locate the genes or the gene loci, and that is why these chromomeres are important. So if you want, you can take a screenshot of this slide and keep it for your reference. You can take this, you can copy it on a piece of paper, or you can take a colored Xerox or a colored printout of this and you can stick it in your NCERT textbook where the chromosomes are being discussed, because all these points are not given in the NCERT in the chapter of cell. So they are given in other chapters, but we are discussing it here today so that you can understand the chapter of cell better, this part given under chromosomes better. So please ensure that you are taking a screenshot of this. Okay, moving on. Now just to quickly recap everything that we've done so far, here you have a metaphasic chromosome. It shows the presence of the sister chromatids which are connected to each other with the centromere. You will notice that each chromatid is going to have subchromatids or subunits, and those subunits are going to be called as the chromonemata, or they can be called as chromonema, chromonema being singular and chromonemata being plural. So you have the chromonemata or the subchromatid. On this side here, you have the metaphasic. On this side here, you have the telophasic chromosome, and in the telophasic chromosome, of course, you will only see one single chromatid. Now, here you have the centromere, which is called as the primary construction, and of course, the primary construction or the centromere will also be present in the telophasic chromosome. Next to the centromere, you are going to have these disc-like structures which are going to be called as the kinetochores, to which the spindle fibers are going to attach during the M phase or during the anaphase. Now, here you can see that there are secondary constructions and secondary constructions sometimes can also be called as the nucleolar organizers. And why are they called as nucleolar organizers? Because they help in the formation of the nucleolus. Okay. Next, beyond the secondary construction, you may sometimes also see a satellite or structures called as satellite. So satellite arm, which is also going to show telomeres at the very tip. So the very tip of the chromosome are structures which are called telomeres, and telomeres prevent the tips of the chromosomes from sticking to each other, but they will help it to stick to the nuclear envelope. And finally, you have the chromomeres. You have the chromomeres, which are small swellings which help the scientist to find or locate the gene locus. So if you want, you can also take a screenshot of this slide, which revised everything that we saw in the previous slide. Okay, so now with this, you have a fair idea about what is the structure of a chromosome. Please remember that none of this is given in the chapter of cell in the NCERT textbook, but in the later year, that is standard 12th, you will be learning this. So it's better to understand this right now so that we have a stronger understanding and grasp over the chapter of cell. And that is why we are doing it right now. So now coming to the parts which are actually there, or the theory part which is actually written in the chapter of cell. So here you can see various cells are shown right here next to me. And in those cells, you will notice that they have said that the cell is in prophase. Now, why is that important? Because if the cell is not in prophase or M phase, there will be no chromosomes, because chromosomes appear only during the M phase. And prophase is the time when the chromosomes will actually start to develop. So here you see the chromosomes in the cell during prophase, and one of the chromosomes has been magnified, one of the chromosomes has been magnified. This chromosome is of course going to comprise of two sister chromatids, which you can see here, the two sister chromatids. They are going to be connected to each other with the help of a centromere. Now, because of this, the chromatid will have subchromatids or chromonema. Now, it is not necessary that each subchromatid or chromonema will be of the same size. One can be bigger and one can be longer and the other one can be shorter. So the shorter one usually is called as the P arm. It is also called as the P arm. And by convention, we say that the longer one is the Q arm. So if you see this in certain MCQs, keep this in mind, that the subchromatids are also referred to as the P arm or the Q arm based on whether they are the shorter subchromatid or the longer subchromatid. Shorter one is usually called as the P arm and the longer one is called as the Q arm. And of course, at the tip of the P arm or the Q arm, the short arm or the long arm, you are going to have the telomeres. You are going to have the telomeres. So now, if this is clear to you, we can speak about the types of chromosomes. So according to the position of the centromere, the chromosomes are going to be classified into four main types. The first type is metacentric. Now, as the name suggests, metacentric basically means that the centromere is located right in the center as far as the chromosome is concerned. So if the centromere is situated in the middle of the chromosome, it is called as metacentric. So this is how the chromosome will appear to be. So if you were to see it under the microscope, this is how the chromosome will look like. As you can see, because of this, the two subunits, the P arm and the Q arm, are actually of the same length because the centromere is right in the center of the chromosome. So in this case, the two arms of the chromosome are nearly equal. They are nearly equal. So now this, what you see on your screen here, children, what you see on your screen here is a metaphasic chromosome. During anaphase, or rather during the prophase, during the metaphase itself, you are going to have the spindle fibers which will attach to its kinetochores. And when that happens, at the time of anaphase, what do you think will happen? The centromere will split and the sister chromatids are going to move towards the opposite poles. The sister chromatids will move towards the opposite poles. So this, dear students, is what is going to happen. So this is at metaphase and this will happen at anaphase. So at metaphase, the chromosome had two sister chromatids attached at the centromere, and now at the time of anaphase, the centromere has split and now we have two chromosomes with one sister chromatid each, or rather only one chromatid. So for the last time, metaphase chromosome, single chromosome, two sister chromatids, single chromosome. At anaphase, we now have two chromosomes. We now have two chromosomes and each one of them is going to be with one chromatid. Okay, now as they move away from the equatorial plate towards the poles, as they are getting pulled, you will notice that they will form this shape. They will form this shape as they are getting pulled. And because both the arms are of the same size, both the chromonimata, both of them, both the chromonema, the subchromatids, they are going to be of the same length. So as they are getting pulled during anaphase, what will be the shape of this chromosome, dear students? As they are getting pulled, what will be the shape? V. So it appears V-shaped during anaphase. So this can be asked to you as a multiple choice question, that what is the shape of the chromosome, or what is the chromosome which shows V shape during anaphase? And the answer is metacentric, because that is the only situation in which both the subchromatids or both the arms are going to be of the same size. So P and Q will be of the same size, P will not be shorter, Q will not be longer, both are going to be of the same size. Okay, so that was metacentric. So the first type of chromosome based on the centromere position. Now, the second type of chromosomes based on the centromere position is going to be submetacentric. So as the name itself suggests, sub means slightly below. So if metacentric was going to be right in the center, the centromere was going to be right in the center, what would be submetacentric? Where the centromere would be slightly away from the center, would be slightly below the center. So if the centromere is situated some distance away from the middle of the chromosome, then such a chromosome will be called as submetacentric. Here is how it will appear. So you will notice that now definitely one arm is going to be shorter and one arm is going to be longer as far as the chromatid is concerned. So one arm of the chromosome is shorter than the other. So you will now have a P arm and a Q arm, a definite P and Q arm based on which one is shorter and which one is longer. Now this is during metaphase. Again, this is going to undergo anaphase. So at the time of anaphase, it will again split and it will get pulled towards the poles. So now when it gets pulled towards the pole, dear children, it will appear as an L. It will appear as a L because both the arms are not going to be of the same size. One will be slightly shorter and the other one will be longer. So if this is going to be slightly shorter, then this will form a L. So in submetacentric chromosomes during anaphase, they will appear L-shaped. So metacentric is going to be V-shaped and submetacentric is going to be L-shaped. Okay. The third type, according to the position of the centromere, is going to be called as acrocentric. Acrocentric. Now the word acros in Latin means towards the tip or tip. A-C-R-O-S basically in Latin means tip. So acrocentric is when the centromere is situated near the end of the chromosome or towards the tip of the chromosome. Towards the tip of the chromosome. Not exactly the tip of the chromosome, but very close to the tip of the chromosome. So here is how a acrocentric chromosome will appear like during metaphase. Of course, one arm of the chromosome is very short as compared to the other arm, which will be much longer. So here the P arm is very short, whereas the Q arm is going to be very long as compared to the P. Now, again, this will be a metaphasic chromosome, which will split into the sister chromatid. The sister chromatids will split into the two separate chromosomes. And now when that happens, these are the two chromosomes that you get. and they will also be pulled towards the opposite poles. They will also be pulled towards the opposite poles. Now, as that happens now. So in the beginning, when both of them were equal, it was V-shaped. Then one of them was slightly shorter. Then it became L. And now this one arm is very short, whereas the other arm is quite long. So now, dear children, we can say that it is J-shaped. So it appears J-shaped during anaphase. So metacentric is V, submetacentric is L, and acrocentric will be J. So this, dear children, brings us to the last type of chromosome based on the centromere position, and that is telocentric. And as the name suggests, you already know telomere means the very tip region of the chromosome. So if the centromere is present at the very tip, we say that it is telocentric. So if the centromere is situated at the tip of the chromosome, it is called as a telocentric chromosome. And here is how a telocentric chromosome will appear to be. Again, this is during metaphase, and if it is going to split during the anaphase, this is what will happen. So it only has one arm and is rod shaped, and this is what is going to happen. So now, as you pull the centromere towards the opposite poles, this is what you will see. And that is why they say that it shows only one arm and is rod shaped. So you have V, L, J and rod shaped. V, L, J and rod shaped. Metacentric is V, submetacentric is L. You will have acrocentric, which appears like J, and finally you are going to have telocentric, which appears as a rod. Some books also mention it as I shape. Okay. The important thing to remember here is that this kind of chromosome, telocentric chromosomes, are completely absent in human beings. So we do not see this in human beings at all. Okay. So here is a picture which you have seen before, and you already know that this picture is a karyotype of humans. So this is basically a picture showing all the human chromosomes during metaphase. And the reason why I'm showing it to you right now is that you can clearly see that we humans do not have any telocentric chromosomes. We do not have telocentric chromosomes. And if you're interested, the metacentric chromosomes in humans are 1, 3, 16, 19, 20. The submetacentric chromosomes are 2, all chromosomes from 4 to 12, 17, 18, and the X chromosomes. The acrocentric chromosomes are 13, 14, 15, 21, 22, and the Y chromosome. And as I mentioned, the telocentric chromosomes are absent in the case of humans. If you so wish, you can take a screenshot of this, and in your leisure time, you can see whether these numbers match or not. But once again, this information is not going to be asked in NEET. This information is not going to be asked in the NEET examination. I am only sharing this with you so that you know that humans have metacentric, submetacentric, acrocentric, but we do not have any telocentric chromosomes. Okay. And here is a chart for your reference about all the things that we have just discussed. So this is something that I definitely want you to take a screenshot of, and I want you to take a printout of this, or I want you to write it down on a piece of paper and stick it in your NCERT textbook where all this theory has been discussed. So here you can clearly see that the first column reads the types of chromosomes are metacentric, submetacentric, acrocentric and telocentric. They have shown you the structures. So in the first one, the centromere is right in the center. So they have shown you the telophasic chromosomes in this diagram. The diagrams shown in this chart are telophasic chromosomes. So you will notice that the first one has the centromere or the primary constriction right in the center. In the second one, it is slightly away from the center. In the third one, it is very close to the tip. And in the fourth one, it is actually at the tip. So those were the positions. And the lengths of the arms nearly equal in metacentric, one arm shorter than the other in submetacentric, one arm very short and the other one quite long in acrocentric, and telocentric of course has only one arm. So V-shaped in metacentric, L-shaped in submetacentric, J-shaped in acrocentric, and rod-shaped in telocentric. So I hope that this chart is now absolutely crystal clear to all of you. Okay. So finally, what does the NCERT say about chromosomes as far as the chapter of cell, the unit of life, is concerned? Once again, I would like to repeat that all these things will be repeated in standard 12th. So right now, there were a lot of things which I discussed which are given in the 12th standard NCERT textbook. And once again, the reason why we did all of this now is so that you have a better understanding of the cell. Okay, so what does the NCERT say? You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers which are called as chromatin. But during different stages of cell division, the cell shows structured chromosomes inside the nucleus, in place of the nucleus. Chromatin contains DNA and some basic proteins called as histones, some other proteins which are not histones, they are called as non-histone proteins, and also it is going to show RNA. A single human cell has approximately 2-meter long thread of DNA which is going to be distributed amongst its 46 chromosomes or 23 pairs. You will study the details of DNA packaging in the form of chromosome in class 12, which I have already done with you in great detail in the chapter of cell division. So you already know everything about chromosomes which is there in standard 12. You already know it while you are in 11th. Every chromosome visible only in dividing cells, because chromosomes are seen only in the M phase. They are not seen in the interphase. So every chromosome visible only in dividing cells essentially has a primary constriction or the centromere, on the sides of which are disc-shaped structures called as kinetochores. And the only simple diagram that they have given in the NCERT is this one. So please remember that what they have shown here is a chromosome. It is a metaphasic chromosome with sister chromatids, and they have mentioned that you can see the kinetochores. So the structures that you see in green in the diagram are the kinetochores. So remember that this diagram can be shown in a multiple choice question, and they will label the kinetochores and ask you to identify the structure. Okay, the next thing is going to be this diagram given in the NCERT. So these are the types of chromosomes based on the position of the centromere. So here clearly there is no telocentric chromosome. We can say that the one which you see right next to my face is acrocentric. The second and the third one, so if you consider the one next to me as the first one, then the ones next to it, these ones can be called as submetacentric, whereas here this one appears to be metacentric. So if we were to see the theory now, which is given in the NCERT, the centromere holds the two chromatids of the chromosome together, of the metaphasic chromosome together. Based on the position of the centromere, the chromosomes can be classified into 4 main types. The metacentric chromosome has middle centromere forming 2 equal arms of the chromosome. The submetacentric chromosome has a centromere slightly away from the middle of the chromosome, resulting in one shorter arm and one longer arm. In the case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short arm and one very long arm. And whereas in the case of a telocentric chromosome, there is a terminal centromere, which is not shown in any of these. Sometimes a few chromosomes have a non-staining secondary constriction at a constant location, and the part or the fragment which is beyond that, as I explained to you, is called as a satellite. So this gives the appearance of a small fragment which is away from the main chromosome, which is called as the satellite. So this, dear children, covers each and every line given in NCERT textbook, in the 11th standard NCERT textbook, about the chapter of cell, the unit of life. So I hope that you have understood everything that we have discussed today and that you have enjoyed today's session. Thank you. God bless. Work hard. Be nice.